Hey everyone, welcome to today's episode of Focal Points. I'm Bram Rector and I'm here with my partner Sam Tabone, who runs Focal Point Partners, an early stage biotech venture capital fund with me. Today's guests are MJ Antonini and Nicki Driscoll, the founders of NeuroBionics. NeuroBionics is a neurotech startup that's making deep brain stimulation minimally invasive through the development of nanofibers that are 10 times thinner than traditional deep brain stimulation leads, which allows them to be inserted endovascularly into the brain. We'll talk more about what all that means later. But first, MJ and Nicki, welcome to the show. Thanks so much for coming on the podcast.
Thanks so much for having us.
Of course. So let's start off with, before we get into all the details about what NeuroBionics is and all the cool technology you guys are building, what was the journey like up to the point where you decided to start working on this? How did you guys get to where you're at now?
Sounds good.
You guys can take that one at a time if you want.
Yeah. I can start. So my background is in mechanical engineering back in France, then switched to neuroscience and neurotechnology by going to the Imperial College London before making my way to the US. And that's where I arrived in Boston about 10 years ago, and I did my PhD in the HST program of Harvard Medical School and MIT. And that's where I joined two of my patients, which was medical world and the engineering world. I joined the lab of Professor Paulina Nikeva, who's developing fibers to interface with the brain, spinal cord, nerves, GI tract, skeletal muscle. And it's throughout my PhD there where we were advancing what we could do with the fibers and how to interface with the body. That we started ideating about translating the portfolio of technology we've been developing at MIT into NeuroBionics, into a company and medical device. And while we were doing this work, Nicki joined as a postdoc, and we started working together. Very quickly realized that we had very, very good chemistry. We were very aligned on our goals and values and aspiration for bringing the technology from academia to industry. And that's how we started NeuroBionics about a year and a half ago.
Amazing.
Yeah.
What about you, Nicki?
Yeah. And so for me, I'm a biomedical engineer by training. I guess this journey really started for me when I was an undergraduate at Brown University, which is one of the universities that was really pioneering in the brain-computer interface space. I kind of serendipitously ended up with Lee Hochberg as an academic advisor when I started at Brown as a freshman. I fell in love with neuroengineering and the idea of technologies to interface with the brain and the nervous system, and really dove into that research while I was there at Brown. And then I decided to continue that research because I loved it so much. So I went and did a PhD in bioengineering at Penn. And there I was really focusing on some of the challenges that had plagued the field at the time, which were devices to interface with the brain tended to be still made out of a lot of rigid metallic and silicon materials, which did not interface well with brain tissue, right? This is the era of the Utah array as the kind of the stronghold of BCI. And so I was really interested in really trying to push the bounds of new types of soft and flexible devices to interface with the brain. And so my PhD work was really focused on some of the material science questions of how we make soft and flexible electronic devices out of new materials. And then about 3 years ago now, 2.5 years ago, I moved up to Boston to do a postdoc with Paulina Nikeva at MIT. And that's how I met MJ and started working with MJ in the lab. I joined that lab as a postdoc because I was so interested in the fiber technology that Paulina's group had been developing for a number of years. It was really innovative and very different than how other folks had approached making devices to interface with the brain, which are, my background was kind of in these traditional thin film microfabrication processes. And the way that fibers are made is like completely different. So that's why I joined the lab as a postdoc. I was also really interested in clinical translation. So I had the opportunity to work really closely with a lot of clinicians while I was at Penn for my PhD. And when I joined Paulina's lab as a postdoc, I met MJ and pretty much right away started working with MJ on projects. And we both were interested in translating technology and really seeing it get beyond the lab and actually make it out into the real world. And so right around the time that I joined the lab, MJ was already gearing up to start NeuroBionics. Right off the bat, he's like, yeah, I really want to start a company with this technology. I think it has a lot of potential. And it really didn't take much to convince me to join the fund. So yeah, it was a pretty natural partnership.
That's awesome. And you mentioned, you got excited about the technology because of the potential that you saw. Obviously, there's a lot of great materials engineering and biomedical engineering that has gone into neurobionics. So I'm curious, MJ and Nikki, if you could talk a little bit about the physical problem that neurobionics is solving, but also the therapeutic problem that you intend to solve too. Hear a little bit about that. That would be great.
Yeah, I'm happy to take this one. So I think the core technology, MJ's introduced it a little bit, are these very thin, flexible fibers. We spent a lot of time identifying what are the core problems that we could solve with that technology. And what we landed on, the one that we're really trying to tackle, is the invasiveness of many of today's neuromodulation therapies. And specifically, if we look at deep brain stimulation, for example, DBS, which has been around since the 90s, and it's used widely for Parkinson's disease, essential tremor, epilepsy, and a lot of other conditions where it's in clinical trials and showing a lot of promise. DBS works really, really well, but its market adoption is abysmal. Really, less than 1% of patients who might actually get therapeutic benefit from DBS actually end up getting it today. And a lot of the reason for that is it's so invasive. It's an open brain surgery. Patient is typically awake during that surgery. And there are also a limited number of functional neurosurgeons across the country that can perform them. And so we kind of saw this problem and said, here's a therapy that works really well, but there's a huge accessibility problem. If we can make this dramatically less invasive, can we substantially improve access to this therapy? And so that's really the problem that we're tackling. So basically, with NeuroBionics, we are trying to deliver DBS into the brain, but do so through a minimally invasive procedure that's kind of like a cardiac stent placement. It's a small incision and a catheter rather than a big open craniotomy.
That's great. Yeah. And I remember when we were talking about this, I was a little astounded at the amount of eligible patients for that would benefit from DBS in different therapeutic areas like Parkinson's and whatnot. And when you told me that less than 1% or something around there actually opt for it, it was very, sad to hear, but also I can understand why. I mean, opting for a craniotomy is no joke, but can you talk to me a little bit about how you're proposing to kind of actually get into the deep brain endovascularly? I mean, this is still a little invasive. I think the common individual may think, okay, that's still invasive if you're going in from the jugular, but maybe talk to us a little bit about that, and also how common endovascular procedures are in other therapeutic areas.
Sounds good. I can speak a little bit about how common endovascular procedure are, and then Nikki can, I think, can take the technical detail of the procedure. We see the like invasiveness of DBS and how decreasing this like barrier to entry, similar to the way endovascular procedure changed the whole field of cardiac procedure. So if you looked at in the 50s and the 60s, there was, I think, about 300, 400,000 procedure every year of cardiac surgery. And then came into play the endovascular approaches to interface with the heart and treating some of the heart issues. That really changed the whole playing field. And now there are about 1.8 million of cardiovascular procedures every year, with a large portion being done endovascularly. So even though we're still invasive, this being minimally invasive really like changed the game. And in the brain, it's very routine for interventional neuroradiologists to place stents and coil to treat aneurysm. They also, every time a patient has a stroke, they get in there and like aspirate or destroy the clots. Overall, this field right now, I think, is the fourth fastest growing medical specialty in the number of clinical claims over the past 4 or 5 years. The number of clinical claims have doubled in the US for this interventional radiology. Nikki, do you want to speak a little bit about the
Yeah. And just to speak to like what this procedure would look like with our device. So essentially there would be small incision in the neck, a catheter that's introduced. As MJ mentioned, this procedure will be quite similar to some of the neurovascular procedures that are done for aneurysm coiling and placing stents for like stenosis or elevated intracranial pressure. And so essentially they'll navigate a catheter and a guide wire up to the target vessel. And then we basically deploy our device from the lumen of a microcatheter. So it actually comes out of a little tube and it expands and anchors itself inside the blood vessel so that all the electrodes on the device are in contact with the vessel wall. And then basically the surgeon will pull out the catheter, guide wire. You're just left with this really thin fiber lead. That comes out and it gets connected to implantable pulse generator or IPG. So that's the electronics unit that houses kind of the stimulation hardware and the battery. And then that gets implanted under the skin, underneath the clavicle. So yeah, there is, a small incision, but it's substantially, less invasive than having the full craniotomy.
Yeah, and so of course you're saving on the craniotomy, the resources required to actually do a craniotomy. That's an inpatient procedure. You need specialists. And so physically, you're really making deep brain stimulation minimally invasive, but more accessible. And then when it comes to actually how that applies to the therapeutic area you're going after with your device, can you talk to us a little bit about that problem and how this is kind of unprecedented in the sense that it hasn't been solved this way before with endovascular DBS?
Yeah, I can do this one.
Yeah, sorry, we're both looking at each other.
Go ahead.
I think so. There have been folks interested in endovascular approaches to the brain for a long time. And honestly, one of the big challenges and one of the big reasons we haven't really seen endovascular DBS yet, it's kind of twofold, but one of the biggest reasons is that to get to these deep brain targets, you have to get into quite small vessels. So basically, blood vessels go everywhere in your brain, but the key is the size. So you tend to have really large open vessels, kind of more on the cortical surface. And as you get deeper and deeper, you get to some much smaller vessels. And it's been traditionally challenging to make devices small enough that you could safely put them in such a small vessel while still having electronics there, right? Still having electrodes that can record and stimulate. And that's really the challenge that we solve with our technology. And that's what our technology does so well. We have this really kind of unique manufacturing approach that allows us to make these very thin fiber-like devices that are super flexible so they can navigate all the twists and curves of the blood vessels, but have electrodes in there that can deliver effective stimulation. So we have some kind of unique approaches to how we make this device that allows us to get into these small vessels. And that's really been the main barrier in preventing and why we haven't really seen endovascular DBS yet. We know there's a lot of folks interested in that direction though. So it's definitely an area that people have been working on and have been interested in for a while.
Gotcha. So it's really the material science innovation is at the core of everything. For someone who is not super familiar with the idea of brain stimulation or brain-computer interfaces, I'm sure a lot of people that are not, intimately familiar with the space, hear about something like NeuroBionics, they immediately think of Neuralink, they might think of, some other big players. What is kind of the canned answer for where NeuroBionics fits into this universe of companies that are trying to put things into your brain? There's many different methodologies, many different indications people are going after, but how do you sort of compare and contrast what you're doing versus some of these larger incumbents that others might be more familiar with?
Yeah, happy to take that one. So there are a number of companies doing building brain implants, and some of them you already named. And I think we can group them with relative to the one that records from the brain and the one that stimulates to the brain. And another axis is where they are placed. So some are on top of the brain, some are and below the skull, some are on the upper cortical layer, but still inserted into the cortical layer, and some go very, very deep into the brain. And with that frame of reference, I think a number of well-known players. So top of the brain and in the skull, you have Motif that is doing stimulation. Very interesting company. There's also Precision Neuroscience as well that have their slit that gets into the upper cortical layer. Then if you go a little bit deeper into the cortical layer, you have Neuralink that you mentioned. There is also one of the pioneer, which is BlackRock Neurotech and the UTRA. And there's also Synchron, which also uses similar endovascular access to the brain and then stays in the superior sagittal sinus and interface with the cortical and sensory cortex. And then deeper into the brain, which is like where we're focusing on, you have mainstay names like Medtronic, Boston Scientific, Abbott, and you also have NeuroPace. And those folks have been the one focusing on deep brain stimulation, which is what we're going after.
Gotcha. Okay, that makes sense. And then what about sort of the non-invasive companies? I mean, there's a lot of hype around things like focused ultrasound at the moment. Maybe that's even used for stroke in some cases. There's obviously something attractive about not having any sort of surgery, even a minimally invasive surgery. What are the limitations of the non-invasive versions?
Yeah, I can speak to this one. So there's kind of two main classes of non-invasive neuromodulation. There's focused ultrasound that you mentioned, and then there's transcranial magnetic stimulation, or TMS. And both of them have some challenges and trade-offs when it comes to depth of penetration versus spatial resolution. So for the therapeutic modality that we're going after, which is deep brain stimulation, you really need to stimulate regions that are deep in the brain with millimeter precision. We'll talk to TMS first. So transcranial magnetic stimulation uses magnetic fields to stimulate the brain, and it's really, really good at stimulating cortical or surface parts of the brain, but it's not so good at penetrating really, really deep. It can get deep, but not with very high spatial resolution. And then if you look at focused ultrasound can get quite deep in the brain, but one of the big challenges is that the skull actually scatters a lot of the signal. And so to get a lot of precision, you either need a very big transducer array that's very powerful, and that's typically how focused ultrasound ablations are done. So you can ablate tissue with focused ultrasound. You can also use a lower power and actually stimulate. To do some of that like precise stimulation, what we've seen with focused ultrasound so far is you typically need like a cranial window. You actually need to make a little window in the skull to get really good precision. So both of these modalities, I think, focused ultrasound is a really interesting one. There's a lot of interest in pushing and advancing that tech forward, but it does run into some fundamental physics limitations when it comes to getting a lot of precision of that signal deep in the brain. And then both of these modalities also right now suffer from kind of bulky hardware that patients often have to go to a specialized clinic to get those therapies. This is not something that they can go home with and have neuromodulation while they're at home in their everyday. So that's really how we see kind of the trade-offs and the pros and cons of these non-invasive versus your kind of implantable devices.
So maybe worth undergoing a minor surgery in exchange for something that's much more precision targeted and you can actually take home with you and kind of, I assume, use indefinitely. There's no real expiration date. Sam, did you have any thoughts on that?
Yeah, I mean, I think anyone that's talked to myself or Brahm for a certain amount of time will hear us use the word precision or a phrase precision neuromedicine quite a bit. And we have actually heard you use precision bioelectronic medicine. And so I was going to ask you, how do you define that? But I think very clearly you kind of just defined it. And so when it comes to thinking about the core technology that NeuroBionics has developed and the device that you're building, you're trying to achieve precision bioelectronic medicine and will with deep brain stimulation. But I'm also curious to talk about the long-term vision and how you can make this precision bioelectronic medicine even better and the different features of what you've built. And so I think you know what I'm alluding to, but I would love to chat about that a little bit.
Yeah, happy to take that one. So the fiber technology behind NeuroBionics, the one that we've been building at MIT for over a decade, was actually developed initially to enable neuroscientists and biomedical researchers to combine several different modalities of brain interfaces into one single device: electrical recording and electrical simulation, which is what we're focusing right now. But it's also combining the ability of delivering optical stimulations and also optical recording, as well as drug and gene delivery. All of that in the context of academic research. So in the lab, we've already validated our ability to do all of that, as well as integrating microelectronics to even add pressure sensing, temperature sensing, and so on. So The long-term vision of NeuroBionics is really to create a platform approach and a platform technology that enable interfacing with the brain across several modalities. And I think succinctly, we're trying to speak the language of a cell. And the cell use different modalities of interfacing. They use electrical signaling, they use chemical signaling, they also use mechanical and optical as well when you think about your photoreceptor in the eye. So the long-term path for NeuroBionic is to integrate each of those modalities that we've validated in the lab into our products. But we are starting with something that is quote unquote like simpler, which is recording and simulation with electrical modalities.
Yeah, no, it was very exciting to me when we first started talking, especially because I'm all about new technologies that can enable a step change in how we treat the brain. But if it can also simultaneously give us richer information about the brain and actually contribute to basic neuroscience, that's amazing. So it sounds like, over time, this platform will be some sort of flywheel in terms of collecting rich data about what's going on in very deep parts of the brain, which is, in my opinion, invaluable. Okay, yeah, that's great. Did you want to say something?
Yeah, I would just say, like, in the conversations that we've had with clinicians, of which we've talked to many, 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 when we start talking about this vision of the multifunctional endovascular lead, they get very excited about that as well, because there's so much we don't know, and it's been hard to get signals out of those parts of the brain and multimodal data. And so there's the possibility to get really interesting data for research and also just to understand the brain better is huge. And so that's something that we are exploring as well. Beyond just a pure therapeutic platform, there's a lot of applications in research here.
Yeah, for sure. And I think, kind of going off of what Brahm was asking you before as well, for the technical audience or non-expert audience, I kind of want to pulse check in terms of the ontology within the neurotech space and clarify to those listening that we're talking about different features and functions that fall into different categories. And so, there's obviously a lot of debate in the literature about what a brain-computer interface is versus maybe, the correct terminology is actually BMI instead of BCI. And so when you're talking about the potential therapeutic use of your technology versus the function to collect information, how are you classifying this in the universe of like neurotech ontology? And what's the easiest way to think about what you're doing?
I think to get back to what I was saying earlier, it all comes down to delivering a signal and delivering a therapy to the brain versus recording from it. And right now, NeuroBionics is highly, highly, highly focused on delivering a therapy, an existing therapy, which is deep brain stimulation using electrical stimulation to provide therapeutic benefits. Recording from the brain also has a lot of uses and have therapeutic benefit, but it's slightly more narrowed in the term of the clinical application and clinical indication that can be used for. So yeah, to sum things up, I think at NeuroBionics we are precision neuromodulation. We don't advertise ourselves as a BMI company or brain machine interface company, really focusing on just providing a therapeutic benefit.
And Nicki, you were mentioning that when you talk to clinicians, they get excited about the research applications, like beyond the therapeutic benefit or the immediate therapeutic benefit. What are some of those research areas that you think people are most excited about?
Yeah, I think there's many. I think so. One place where we've really heard a lot of excitement about is like we're planning to go into the brain through the venous system. Right now, there are a lot of interventions, endovascular interventions in the brain are done on the artery side. There are fewer done in the venous system. And there's a lot of interest in trying to understand how certain disorders may actually stem from disorders that come from venous circulation. And so we've had clinicians say, like, even if you just had like pressure and flow sensing on that fiber while it's there recording, stimulating, like that would be invaluable data that we don't have access to today. And we would learn so much about brain circulation. It turns out that venous circulation in the brain has been kind of ignored for a really long time. And now there's increasing evidence and a lot of hypotheses around certain disorders that may stem from kind of disorders of circulation. So there's a lot of interest there in terms of the vascular kind of function, but then also just neural function, right? If we're recording neural data and neural activity in some of these brain targets that are a little harder to reach with traditional DBS devices, there's a lot of interesting applications there because, I think a lot of the data that we get from human patients who advance the field of research forward, a lot of that ends up coming from epilepsy patients who have stereo EEG. And that's been incredibly useful in advancing the field. But there's kind of a caveat here where a lot of the data that you get from patients that have epilepsy may not represent the normal healthy brain. And a patient that's having DBS who maybe had a stroke maybe has slightly more typical brain function in different regions. And so we might get access to some really interesting data just having the ability to record in those regions.
I see. And at the risk of getting into the weeds, or I guess maybe the veins, you mentioned that most endovascular procedures happen on the arterial side, but you guys are going through the venous side. Why did you make that decision to go on that side that's less commonly used?
It's really a safety question, actually. So there's a lot of reasons. The veins are a much lower pressure system than arteries. They're more compliant. It is safer to leave a device in a vein than it is an artery in terms of the risk of thrombosis or stroke, but also just your device being able to stay anchored and not dislodge over time. And another reason is that arteries tend to have a little bit more smooth muscle around the outside of the vessel. And so if you had a device there that's stimulating, there's a slightly higher chance that you may actually cause vasoconstriction, which means the vessel to constrict a little bit. Veins have significantly less smooth muscle and a very thin vessel wall. So when you have a device that's in a vein, you can actually stimulate brain tissue quite easily. And the vein doesn't really block the transduction of your signal very much. And you don't have that issue of vasoconstriction the same way that you might have in arteries.
Interesting. Just to continue on the research thread, I know that NeuroBionics is very early, but you have done a number of studies in rodent models. And if I remember correctly, I don't have it in front of me, but there was something that was about stimulating maybe the GI tract or something like that. Can you talk about some of these interesting studies you've done in animal models and why they're interesting and if they'll ever make it to humans?
Yeah, I can take this one. So the lab of Professor Polina Nikeva has done a lot of work in the fiber technology space. So we've, over the past 12 years now, we've worked with mice, rats, pigs, and also non-human primates in different spaces. So the brain, most of the work has been done directly into the brain, but there's also some done directly into the nerves, some into the spinal cord, and finally the one you mentioned into the GI tract. One of the recent paper from the lab from last year was using the fiber to both monitor the activity into the brain and the activity into the GI tract at the same time, because I'm sure you're aware of that. There's been an explosion of interest into the gut-brain axis and how some of the disorder we thought were stemming from the brain might actually come from the gut. So and the tools to interface with the gut are not there. So having something that is really, really long, very, very flexible, and that has the ability to record and modulate in the brain was really, I wouldn't say easy, but was possible to translate that into the GI system. And that's some of the work that we've done in the GI tract. We have a couple more work on that in that space that should be published soon as well. So I can't say too much, but this is also some additional work on the GI tract.
And is that mostly? I know you can't say too much. Is that mostly on recording, or is there stimulation as well?
Simulation.
Okay, I see.
So both work had. So one was recording and stimulation and drug delivery. The one on rodents, and the most recent one that is not out yet was electrical stimulation in the GI tract.
Very interesting. Okay, Sam, I've been hogging the mic. I'm sure you have an intelligent question to ask.
Yeah, I think I have a very technical question that I'm going to save after we kind of work through the first set of questions here, just in the context of walking through a hypothetical situation in which a stroke patient who's recovering receives treatment with your device. As far as I know, and I may be wrong, but the best treatments that exist for stroke right now, there's acute intervention, whether that's surgical or with a tissue plasminogen activator. And then post stroke, chronically, you have physical rehabilitation and basically just therapy. So correct me if I'm wrong, but I think that's all that exists. And in terms of a patient's journey, at what point would they interface with the NeuroBionics device?
Yeah, I'm happy to take this one. So, and just to clarify, like, I don't think we were talking about DBS, but yeah, we are looking at DBS for stroke rehab, which is kind of a new direction. So, to your point, Samantha, you're absolutely right. Today, when a patient has a stroke, they typically come to the hospital and they have emergency care, which is often a combination of TPA and today more and more a procedure that's called thrombectomy, which is actually navigating a catheter up to the clot that's in the brain and aspirating it out. And that tends to get the best outcomes. So these patients are already having a neuroendovascular procedure to remove the clot from the stroke. And what typically happens is they will have some acute care in the hospital, and then they'll go home and they'll do a combination of physical and occupational rehabilitation to try to recover some of the function that they may have lost from the stroke. And today, in a stroke patient's kind of pathway, a lot of times they'll end up recovering a certain amount of function, usually in the first several months after the stroke, and then they tend to kind of plateau. And beyond there, they don't tend to see a lot of improvements or gains. And so if they have some lingering deficit, like a limb weakness or motor disability, they tend to not have a lot of options for how they can rehab that because they've sort of plateaued and there's not really much else that we can do for them. More recently, there's been some really interesting work showing that if you deliver stimulation to a particular region of the brain in the cerebellum, you can enhance something called neuroplasticity, which is the kind of the brain's ability to sort of rewire itself after injuries. And you actually can help these patients recover more function. And so, where this patient might interact with our device, the way that we see it is it could be anywhere from about 2 weeks post stroke when their stroke risk has gone down and they're kind of back to their sort of pre stroke hemostasis. Up to actually several years after stroke. So there's been some evidence that some patients who maybe have already gone through the whole process, done some rehab, they've had some recovery and they plateaued, they could go through another course of rehab therapy with DBS and actually see significant improvements. And so this is really exciting to us because this means that we could potentially have a pretty wide therapeutic window. We could intervene with these patients as early as 2 weeks after the stroke or as long as maybe even several years after the stroke and still have some therapeutic effect for these patients in helping them recover more function. So that's really the pathway with neurobionics.
Yeah, and to kind of dive a little bit more into the neuroscience a little bit, and I know that this is now speculative discussion when I start asking about things like by which mechanism do you think the stimulation will actually induce plasticity? Is that a global functional connectivity level? Is it more enhanced if you're actually stimulating closer to a specific brain tissue type? What are your general thoughts on that type of I guess musing at this stage? You can't really, prove anything, of course, but I think it's
Yeah,
fun to think about.
yeah, absolutely. So I will say, like, we stand on the shoulders of giants, and we are building this upon a lot of research work that has come before us. And there's been a lot of research into what this potential mechanism is. And a lot of what we're basing this on is some work that was done by Andre Machado and a group at the Cleveland Clinic. They spent decades showing this mechanism in rodents and then moved into humans and showed that it worked in a phase 1 trial. What the mechanism is proposed to be is essentially you're stimulating something called the dentato-thalamocortical pathway. I hope I pronounced that correctly. It's a pathway that goes from the dentate nucleus through several deep brain nuclei, and then there's a white matter tract that projects all the way up to the cortex. And what we believe the mechanism is when you stimulate that that nuclei in the cerebellum, you activate this whole pathway, and activating this pathway actually causes the plasticity in the cortex to increase. So I don't fully know the mechanism of exactly how it increases plasticity, but we basically know that activating this whole pathway has been shown to enhance plasticity, and that's what's really leading to this therapeutic effect.
That's quite exciting. And I'm not sure if I've actually ever asked you this or not, but do you have any hypothesis on whether treating someone post-hemorrhagic or post-ischemic stroke will differ?
Yeah, so basically, we think that this is going to be most effective for post-ischemic stroke. Patients that have had a hemorrhagic stroke tend to be at elevated risk of additional strokes. And so they actually might not be a great candidate for this type of therapy because their stroke burden, their risk of follow on stroke remains high. So that's actually one challenge. This is a little bit better suited to ischemic stroke.
I see. Yeah. And, just kind of continuing on this conversation of the ideal, I guess, mechanism by which you stimulate the deep brain, I have to ask because we have investments in other areas like this, but in a future in which we can deliver drugs to the brain through your device, perhaps, do you foreshadow like maybe delivery of small trace amounts of a neuroplastogen in conjunction with this, enhancing plasticity even further and showing an even more dramatic effect in stroke recovery?
Yeah, I can take this one. So, the answer is yes, definitely. As I was alluding to earlier, our ability to deliver drugs and gene through the fiber was validated in lab, and we definitely have planned to integrate that into our fiber technology platform through the endovascular access. And that opens the door to not only to improve the outcomes to some of the indication we can pursue, but also opens the door to like a lot of more interest, like not more interest, like even more like applicable indication that combine drug delivery and electrical delivery or electrical recording, both in the brain but also like in the periphery. So there is it opens the door to many indication, which is really exciting for us, and that's definitely part of our plan.
Yeah, and
Are
there's
you?
absolutely evidence. Oh, I'm sorry.
No,
There's
no, go
absolutely
ahead.
evidence that delivering certain agents that can enhance neuroplasticity with stimulation can have even greater outcomes. But that's mostly been done in the research space, and I don't know if that's been tested in humans yet, but super exciting to see the potential of translating that to human patients.
Yeah,
Yes.
a clear example I think would be a paper. So deep brain stimulation I mean, until now has been done with electrical stimulation, and there's been in research a lot of interest in refining the specificity of deep brain stimulation by using optical stimulation, and there has been a lot of work in that space. And there is a key paper from a couple of years ago that showed that you could reproduce the therapeutic effect of optical stimulation that would target a specific cell type by combining drug delivery and electrical stimulation. Basically, the drug delivery would like force the cell types that you do not want to activate and like quiet them down, and then the electrical stimulation would then activate only the pathway that you are interested in. So that there's a lot of way by adding several modalities together, you can leverage the advantages of each of single modalities.
I mean, the possibilities seem endless. I have a few friends who are neuroscientists that always talk about their dream of being able to selectively deliver drugs to the striatum, for example. And so, yeah, it's very exciting. And I guess for those listening who are curious, I mean, within what you can actually share, the material that you're using for this fiber, going back to the fiber, we're talking about delivering stimulation, but in theory also delivering drugs. Can you talk about how that would work and if you could actually, I don't know if multiplex is the right word in this context, but actually maybe collect different bits of information simultaneously?
Yeah, I can speak to that. So the manufacturing process we are leveraging basically enable us to fabricate and manufacture everything at a microscopic scale. So at a centimeter scale, we fabricate what we call a preform that has electrical, optical, chemical modalities. So for chemical modalities, it's just a channel. Electrical, those are going to be the wires. Optical is an optical waveguide. And then we effectively shrink everything down to a microscopic scale. And the material platform is a polymer platform. So we obtain those very, very thin fibers that are very flexible, made out of polymers, and that have microscopic channels for drug delivery. But at the same time, you have those microscopic electrodes for recording and stimulation.
Gotcha. No, thank you for explaining that. It's very interesting that all of that can happen in one, several micrometer thin fiber. I have a highly speculative, futuristic question.
We love that.
So, yeah, I mean, we live in LA, and it's very obvious that a lot of people get surgery that's not necessary, not for any medical reason, I'm curious to know what you guys think about a future world where people potentially get, various brain implants, not for any particular medical purpose, but to maybe enhance some type of neural function, maybe increase their IQ, maybe something else. Do you think we ever get there, or is that just a bunch of tech accelerationists in the Bay Area that want that to happen, but it's never going to happen?
I would say a bit of both, and I can start, and then Nicki, you can like share your perspective. So those are really like individual perspective. I don't think we've had a considered
fair
thoughts on that.
Well, if you just think about it, I mean, some of the procedures that people get purely for cosmetic purposes seem much more invasive than an endovascular, brain fiber. Yeah. So I don't know.
And I think, so the short answer, I think we're going to get there eventually. I think the way to get there is going to be really, really, really key. Not only because of safety and efficacy and all the issues and like whether it can work, but also the ethical implication of those augmentation. And I think because those surgeries and those therapies are going to be costly, there's going to be a barrier to entry, and making sure that they are accessible to everyone so that it doesn't like further increase the gap between people that have like some advantages in life and people that are less disadvantaged. So it's really, there's going to be a whole host of neuroethical consideration that needs to be really, really well laid out at a policy level, but also like at the individual company level.
Yeah, I totally agree with that perspective. And I think there is a chance we get there, but I think we're a ways away from people opting in for non-medical implants like this. Even a device like ours is not something that you might want to do for just like it's not medically necessary. I think we're quite a ways away from that, but I do think it might happen eventually. And I think, to MJ's point, absolutely, then it gets into some really challenging questions of neuroethics in terms of, are these actually going to be augmenting people's abilities or their cognition or anything like that? Then it does become challenging. And there's a lot of really fantastic discourse about this and these neuroethical questions amongst many people in neurotech. All the giants of neurotech are keeping this in mind because it's such a new field and new technology. And anytime you have new technology, it always brings up some interesting new ethical questions that need to be explored. So I think, to MJ's point, it'll require just being really thoughtful and making sure that if we do get to that point where folks can just opt in to have something like this, that it's done kind of in the right way and doesn't kind of increase disparity.
That makes sense. I mean, has there even been any research that shows that this type of stimulation could even increase IQ or something like that, or is that just kind of sci-fi?
There is some very limited evidence that certain types of stimulation can enhance memory, but it's mostly been done in the context of Alzheimer's disease and folks who are already losing memory.
Right, people that are
to my knowledge, I don't know that there's a lot of evidence that you can really enhance a healthy person with normal cognition.
Yeah, that's fair enough. That's probably for the best if it turns out to be the case. Okay, so going back from the future to now, I mean, where is NeuroBionics at this point in time? What does the next year, next 3 years, maybe next 5 years look like for you guys? Like, what's the timeline in the near to medium term for y'all?
Yeah, so We are, so we spent a lot of MIT full time in January 1st, 2024. So we are quite young as a company, but we have behind us a decade of technological innovation. Right now, what we're focusing on is on the productization of the technology and the translation of this scientific research and technological advancement into a clinical device that will be deployed through the vascular system and interface from inside the brain, from inside the blood vessel. And that goes along with validation of the safety profile and the efficacy in animals. So over the next year and a half, 2 years is going to be our preclinical studies to really validate what we want to do. And then after that, it will be moving towards first in human and early feasibility studies. And finally, a phase 3, which can be a pivotal trial. That's probably going to be like 5-6 years from now.
Gotcha. Okay. Sam, I know that we have like 2 more questions and then we can, get out of here and you guys can get back to putting wires in people's brains. Sam, did you want to ask the question?
Yeah, I mean, the preamble for my question is that I've spent a lot of time with technical founders in and outside of academia. And part of my excitement in funding neuro companies, whether that be on the pharmacologic side or the devices side, is that we're really translating new basic insights, new engineering into the real world and accelerating that at a very high rate. I also feel like there is an innovation crisis in a way in academia, but coming from one of the most famous technical universities in the world and spinning out of that, I would like your perspective on how you've experienced the speed of research in academia versus now in a startup, what the pros and cons are, and I guess how you compare and contrast that as founders.
Yeah, there's a lot there. I can speak a little bit and then I'll let MJ chime in. So the speed is very, very different. Everything moves way faster in a startup because it has to. You have to really deliver quickly and hit your milestones and move forward. And once you leave the university and you're building a company, you have to raise money to do that, and then you have to deliver and hit your milestones. And so I think what's really key is having reached a point of development in the academic setting where there's not a lot of additional quote unquote R&D to do. There's maybe a little bit, but it's not going to take you, 5 more years of research before you have something that can become a product. And so I think that's really key for folks in academia who are thinking about starting companies, is really thinking about when is the right time, and how mature do you want this technology to be before you leave academia and start a company. But I totally agree with you that there's so much incredible innovation in academia that doesn't make it out. It doesn't make it into the real world, and that's a real shame. And that's a lot of the challenge is just finding academics or others who are willing to take that tech and translate it through, what we call the valley of death, actually really getting something from academia and making it into a real company and a real product that is going to work. Yeah, and just one last thing to add, you mentioned the university that we've spun out of, MIT. We've been incredibly fortunate to come from the ecosystem of MIT, which does an incredible job of supporting entrepreneurs and giving them the resources to learn what it takes to start and launch a company and really set them up for success. And we've benefited so much from that ecosystem and from so many incredible mentors who've been willing to give us advice and help us along the way.
Yeah. So if I can add to that, I think the difference between academia and entrepreneurship, or at least like in our experience, was that in academia it was really focusing on the science and the science for the sake of the science. And get to a point where all the scientific questions were answered, and we had very good confidence that those problems were solved. And now it's going to be a engineering and business and regulatory and clinical and reimbursement problem. And so once we're lucky enough to be at a point where the science was like mostly done, and now we are taking that out into the real world and thinking about all of the other problems. And it was like, okay, cool, science is done. And back then we were like, oh, the hard part has been done because so much has went through. except that it wasn't really the case, because now we have to think about, okay, do we have a business case? Do we have a clinical case? Do we have the regulatory challenges? What are the risks there? What are going to be the reimbursement risk? What's going to be the market adoption? And so on. So because you also have so many other dimensions you can think about, things move a lot faster. But more than anything, I think the thing we benefited the most was people's willingness to help each other in the entrepreneurial world. Like, we've received mentorship and received advice, and like people spending an inordinate amount of time and energy to help us just because they went through the same process and they know that starting a company is like challenging, and there is a philosophy of like paying it forward that's like really, really embedded in the entrepreneurial ecosystem. And from an academic background, like, I was really, really, really shocked because my idea of industry was like, oh, everyone is against each other, and like everyone's like have their business incentive and they're all fighting each other. At least at our stage in like startup world, that's the complete opposite. Everyone is like going out of their way to help each other.
Yeah, I think that the sort of pay it forward mentality that you described does surprise a lot of people that come from academia. I've heard other folks say that. So that's something that maybe surprised you in a good way. Is there something that maybe surprised you in a bad way, that maybe you thought, oh, this part of business or being a founder is going to be easy, but it was actually way harder than you thought?
I wouldn't say in a bad way, but we're surprised like every day. I think like the like a year and a half ago, when with Nikki we were like, okay, we're incorporating NeuroBionics, and now we're gonna like take the steps to like make it into a reality. Like every day we realize like how much work there is to be done, and like a whole new axis of hurdle we have to like work through. So I wouldn't say it's in the bad way. It's just we were very naive when we started in like what it takes to bring class 3 medical device and active implantable medical device to the market. And every day we're learning like how interesting but challenging the path will be. I don't know, Nikki, do you have anything else with that?
I mean, I think you said it really well. Yeah, I can't think of anything in particular that like doctor surprised us in a bad way, but yeah, there's just been a lot to learn. And like little things that we didn't think about, like, okay, how are we going to set up our HR policies? Little things that we just like didn't think about that all needed to get done and needed to be organized and put in place. And setting up our hiring strategy and how we're going to run a hiring process. So it's been, so much to learn. And I think we've both really, really enjoyed that process of learning what it takes to run a startup and to build a good team. And so it's been a lot of fun, and there's been a lot to learn and a lot of personal growth.
Yeah, it's definitely a lot to learn and a lot of work, but as far as I can tell, both of you guys are doing a great job of absorbing it all and executing on it. So kudos to you. If only everyone could be so, neuroplastic, and the world would be a better place. Well, you guys have given us so much of your time. I don't have any other questions. I don't think you do either, Sam. Is there anything else that you guys want to leave the audience with? Maybe you could, reference your website for people that want to check you out. Maybe, I don't know, if you're looking to hire anyone, you could talk about those roles that you're looking to fill in case some, highly motivated engineer happens to be listening to this. But any place you want to leave this, I'm open for.
Sounds good. Yeah, I mean, like a couple of like parting thoughts. The first one is like check out our LinkedIn and our website, neurobionics.io. We're going to be posting a couple of job description, one focusing on a mechanical engineer with expertise in endovascular space, and the other one will be on research R&D development with a neuromodulation expertise. So be on the lookout for those. But more than anything, I'd encourage everyone from academia to like take the leap, and because it is a leap of faith, and going to the entrepreneur world, it's 1,000 times more rewarding than what I expected, and I thought I knew, and it's even better than expected.
That's amazing.
I think that's a great note to end on.
Yeah, encouraging more competition. I like it. Cool. Well, yeah, MJ and Nicki, You guys have been great guests, and thank you so much for doing the work that you're doing. The world needs it, and thank you for letting Sam and Focal Point Partners and I to be on your cap table. It's been, really cool seeing you guys make the progress you have so far, and looking forward to seeing what happens in the next 10 years. But we'll do this again at the IPO. And until then, check out neurobionics.com or .io, whatever the website is.
.io.
Thanks, guys. Bye.